Every second, something amazing is happening within our cell. Microtubules spans wide roads for tiny motor proteins to walk along, carrying cargo around. Today, we're exploring the wonders of our cytoskeleton. The cytoskeleton is built from three main types of protein filaments. Microfilaments have a diameter of 6 nanometer, it in charge of controlling cell shape. Intermediate filaments have a diameter of 10 nanometers, its role is to provide mechanical strength. Microtubules have a diameter of 25 nanometers, it acts as highways to transport cargo around the cell. Together, these fibers give cells strength, structure, and the ability to move. Microfilaments are the smallest protein filaments. They're made from actin, a globular protein that assembles into long, helical filaments that come bounded to an ATP molecule. Actin filaments are polar. They have a plus end and a minus end. New actin molecules bound to ATP is added to the plus end and is hydrolyzed to ADP upon addition. The minus end thus only include actin bounded to ADP, which is unstable. This creates a process called treadmilling, where the filament appears to move, adding actin at one end and losing it at the other. Actin is usually concentrated beneath the plasma membrane in a region called the cell cortex, where it controls the cell's outer shape. It nucleates near the membrane using a complex called ARP2/3, which we won't go into here. Because of this arrangement, actin can rapidly reshape the cell surface, allowing phagocytes to engulf pathogens and epithelial cells to maintain firm, stable shapes. Actin's dynamic nature also drives cytokinesis, where actin and myosin form a contractile ring that pinches the cell in two during division. Next are the intermediate filaments. They're rope-like fibers made from different proteins depending on the cell type. Keratin in skin, desmin in muscle, and lamins in the nucleus. Their main role is to provide mechanical strength against tension, preventing cells from tearing when stretched. They're far more stable than actin or microtubules, and they don't treadmill. They form a network around the nucleus called the nuclear lamina, providing support, and they also play a key role in anchoring organelles. Finally, the microtubules, the largest filaments, about 25 nanometers in diameter. They're hollow tubes made from alpha and beta tubulin dimers, which line up into 13 parallel protofilaments to form a cylinder. Like actin, microtubules are polar. The beta tubulin end is the plus end, which grows and shrinks quickly. The alpha tubulin end is the minus end, which grows slowly and is usually anchored near the nucleus. Microtubules, like microfilaments, can grow and shrink, however. Instead of ATP, it uses GDP. Instead of treadmilling, microtubules mostly just grow and shrink instead of moving. An interesting mechanism called a GTP cap can form when the rate of GTP addition is high enough that it forms an area with mainly tubulin bound to GTP. This stabilizes the microtubule and allows growing. Without the cap, the tubule bends and collapses. Microtubules grow out from structures called microtubule organizing centers, or MTOCs. The main one in animal cells is the centrosome, located near the nucleus, containing a pair of centrioles, arranged perpendicular to each other, surrounded by a matrix that nucleates new microtubules. From here, microtubules radiate outward, acting as tracks for transport and structural supports. They also form key structures including the spindle fibers that separate chromosomes during mitosis, and the cilia and flagella that move cells or fluid. A highway made from the cytoskeleton won't function without the cars driving on them. And here is where motor proteins come in, the cargo-carrying machines that runs on ATP. Kinesins move along microtubules toward the plus end, away from the nucleus. They transport cargo such as secretory vesicles and neurotransmitters during axonal transport. Each kinesin molecule has two heads that act like legs and a tail that holds the cargo. One head binds ATP, the other ADP. When ATP is hydrolyzed, it releases energy that propels the head forward. An 8 nanometer step, the length of a tubulin dimer costing 1 ATP per step. Dyneins move in the opposite direction toward the minus end, back toward the nucleus. They're much larger and more complex, needing a helper complex called dynactin to attach to cargo. Rather than walking neatly, dynein moves more like pulling a sled. It carries lysosomes, damaged organelles, and drives the bending motion of cilia and flagella. In summary, kinesins and dyneins travel along the microtubules. Dyneins carries things towards the nucleus whilst kinesins away. Myosins are the motors that walk along actin or microfilaments instead of microtubules. There are many types. For example, myosin 2 powers muscle contraction and the contractile ring in cytokinesis. Myosin 5 transports vesicles and helps reshape the cell membrane. All myosins use ATP hydrolysis to produce force and movement, controlling fine-scale shape changes and local transport within the cell. So the cytoskeleton isn't a rigid frame. It's a living, dynamic system. Microfilaments shape and move the cell. Intermediate filaments give it strength and resilience, and microtubules organize the cell's internal layout and provide transport routes. Powered by motor proteins like kinesin, dynein, and myosin, this molecular framework keeps the cell structured, mobile, and alive. A microscopic world of constant motion and coordination. Thanks for watching. Don't forget to subscribe if you enjoy these videos.